Hi, everyone. I'm excited to do an interview with Roger Deutsch, the CEO of Cell Science Systems, who does Alcat testing. And I have a lot of clients who've done Alcat testing, and a lot of people have questions. And I had a phone call with Roger last week. I learned some new things, and I thought if he was willing to let me interview him, this would be really informative to my clients and maybe other people on YouTube that find this video and have questions about allergy testing, especially serum allergy testing. So Roger, can you introduce yourself, talk about your history with, you've been doing this a long time, and also the history of Cell Science and how you guys started doing something different with allergy testing?
Absolutely, thanks. First of all, thanks a lot for having me on your webinar. So I've been involved with this food sensitivity testing company and this Alcat test since 1986. And how I got here was that growing up, I had very serious allergic issues. And at that time, mid-late 60s, well, early 60s through that decade, I was looking for ways to treat it, and I went to the local allergist. And I grew up in the Northeast, and we had hay fever season in late summer, spring, and that was very difficult for me. To make a long story short, although conventional allergy treatments were beneficial for a great many people, they didn't benefit me. And in those days, also the prescription of these first generation of antihistamines would just knock you out, and I couldn't tolerate that. So I spent a lot of time and a lot of effort and many miles of travel to try and figure out how to understand allergy for myself, for my own benefit. I took some detours and I got into the oil business, and for a period of time, late 70s, early 80s, and I found that very exciting, and it was a good activity for a young guy. But I wanted to get back to what I was really interested in was natural health. So I had pursued investigating what caused these allergic symptoms, and one of the things along the way was that diet. I was suggested by some, I was living in Europe for a while in the early 70s. I went to study Oriental medicine in the UK in the early 70s, and had a common cold, which seemed kind of stubborn. And one of the Australian naturopaths said, look, you need to try a mucusless diet. And I thought, what is a mucusless diet? Nowadays, probably people would refer to that as a anti-inflammatory diet. It means cutting out things like all wheat and all grains and dairy, and eating fresh fruits and vegetables primarily. So I did that, and I was just amazed at the way I felt. I had already switched over to become a vegetarian some years before, and I found that beneficial. I was definitely into eating healthy foods, meaning fresh foods, not processed foods, a lot of fruits and vegetables, and some whole grains and so forth, nuts, seeds, etc., and a little bit of dairy. But that experience opened up my eyes to how profoundly beneficial the right kind of diet will be. So fast forward a few years, I needed to get down to earning a living, and so I entered into the oil brokerage business with some colleagues, and we developed what became the largest oil brokerage business in the world. And I used to communicate with people in Houston every day for 7 years. And it was a great activity, and we made, it was very successful, but I felt that was not exactly what I enjoyed doing. I wanted to get back to pursuing anything and studying about healthy living and how the body works in a more profound way. So I heard about some folks that had been experimenting with this food sensitivity testing, and they had a patent issued, and they didn't have any wherewithal to commercialize it. And they, through an intermediary, asked me if I'd be interested in starting up a business and making something of it. And it just hit me. I had already been out of the oil business. I'd moved to California. I was pursuing my lifelong dream of swimming in the ocean every day and playing tennis every day. And and doing some other healthy activities, eating organic and having a great old time. But it was looking around because I was a tad bored. So in 1986, I came out here. I brought my son with me and my then wife, and Jason was just about 9 months old. This is kind of interesting in relation to the story because Jason, as you can understand, given my mindset, he was born at home by natural childbirth and ate only organic food. He was breastfed for the first part of his life, then was beginning to eat some foods that were all organic and healthy, but he was the healthiest kid you can imagine, but then he got this rash all over his body. And that coincided with the time we came out to Florida to meet with these folks that had just gotten the patent issued. And I said, well, what do we do about Jason here? If your test works, what can we do? And so we ran a test on Jason and found out that he came up, his blood indicated a strong reaction, something he'd been eating quite a lot of. We took him off that and he cleared up. Then I did my due diligence, speaking to various physicians, and I looked into this technology, and I felt that there was the fundamentals of something quite profound. And of course, it was completely aligned with my particular interest in food and health and trying to understand how the right food or the wrong food plays such an important role in terms of a person's health. So I got involved by starting up a business in 1986, and that's been my exclusive activity for the most part since then. So it's coming in, actually this is about the 34th year that I've been doing this. So I find it totally interesting and keep learning stuff, new stuff every day.
And what was it that he was reacting to?
Green peas.
Green peas, okay.
Yeah.
So that's interesting because green peas, I didn't tell you this the other day, but I'll tell you what happened on my first Alcat experience. But green peas is on my mild list right now. And I was having a green pea protein shake powder every morning with vanilla flavored, which was also on my list. So I had to scrap the morning routine I've been doing for about the last 2 years. But one thing I didn't tell you last week, which has to do kind of with your story, was the very first time I was introduced to Alcat was 15 years ago. And a chiropractor in the Houston area, someone else had told me about Alcat. She had a good experience, so I had it done. I had done the platinum test because of my previous history was I was very autoimmune, chronic fatigue, fibromyalgia, allergies, asthma. You know, I was on 4 inhalers a day, including emergency ones to make sure I would like stay alive. Couldn't exercise, you know, just having a lot of problems. Well, the interesting thing that happened to me was that by the time this was 15 years ago, I had already went on the, you know, back then, remember 20 years ago, there was rice cakes. That was the gluten-free thing at the grocery store compared to now. And but I was already corn-free, dairy-free, wheat-free, gluten-free, and didn't have peanuts and didn't have already a lot of things. And I still couldn't get there. You know, there was still some things that were challenging me. So anyway, when I did the test that this chiropractor says, he was also a dark field blood guy. And he says, you know, your blood is very inflamed, and it was a different thing than Alcat, but he had looked at my blood and said it was, you know, had other issues with that dark field looks at. And so his recommendation was, we'll go home and, you know, start desensitizing by having 1 vegetable a day, and you can have olive oil and lemon, but you can't have any, you know, don't have anything else until we get the results. So I did that, and about the 4th day, I was so sick, like had a total relapse of my fibromyalgia symptoms. And I called him, and he said, well, that's not possible. And I had followed instructions. 1 whole day I had cauliflower, and 1 whole day I had broccoli, and 1 whole day I had green beans, and just seasoned it with salt, lemon, and olive oil. Well, on that test I got back 15 years ago, on my severe list was lemons, olive oil, which were the 2 things that he said do as much as you want with your 1 vegetable as seasoning. And I just had a normal amount. You can't really do too much olive oil, you know, it's just enough to saute or, you know, put it in the oven. And and almonds. I'll never forget, those were my 3 severes, and I was eating those 3 things every day. And they weren't the typical things that people say to avoid. They were my list. Okay, fast forward 15 years, I just had it done again. And almonds is still on my severe list. And, after 15 years, I kind of had decided it's not bothering me, but I was doing some like paleo, different kinds of alternative pancake mix or, things made out of almond flour. And I was also doing almond milk because I haven't had cow dairy in over 20 years.
Hmm.
So I've depended on Alcat to steer me in the right direction because I'm also a blood type theory kind of person. If someone doesn't get Alcat done, I generally go on that premise for myself. It helped me a lot, except for that little bit that I couldn't identify because with my blood type, almonds, lemons, and olive oil are okay. They're considered neutral foods. So anyway, that's kind of my story how I got into Alcat. Once I was able to really identify and fine tune those things that I was avoiding, I got the rest of the way. And the interesting thing was, and we're going to talk about this because it's one of my questions, is that a few of the things that I had been completely avoiding, like corn, showed non-reactive. And I still don't eat too much corn because of GMO and a lot of other reasons, but I will have it occasionally. And it showed up again this time on my recent test a few weeks ago as
Hmm.
but it has always shown up as neutral. But I just have this thing in my head, I think, that it's one of those things that I shouldn't have too much of. So that was my experience. You had told me on the phone, and I think now's a good time to share this, unless you want to share something else anecdotal. I have the book that you guys, I found out about from someone at Alcat about your hidden food allergies or making you fat. And I know that for me, a lot of my clients are autoimmune. That's my thing, is autoimmune. Weight loss is not my number one objective with clients, but sometimes it's a nice side benefit. I did have a client of mine that just had the Alcat with the free promotional CNA testing done a month ago. He came in Friday after we talked, and he had lost 12 pounds since last month without doing anything different except avoiding those inflammatory foods. So you told me a great story last week about an actor that will be nameless. Can you share that story or any other kind of story that has to do with someone being able to recover with
Lisa, please call extension 100.
Sure. Well, the the actor story is kind of interesting. So this is, of course, I can't share his name because I don't have permission to do that, and it's private. But it's an A-list actor that everybody knows, and he had to prepare for a part in which he played a character who was very overweight and very unhealthy, or relatively overweight and unhealthy. And this was, as many A-list actors are, pretty healthy and fairly fit, very fit actually. So he had 2 weeks to gain 30 pounds. So we came to a doctor who told him, well, let's do the Alcat test. And for 2 weeks, just eat the red foods, meaning red foods are the ones that are strong reactive ones. And so he did that. And sure enough, in 2 weeks, he gained a little over 30 pounds. And his part also required that he looked ill, and he was pretty ill. So, that's not the way we recommend to use the Alcat test, but that showed how powerful the wrong foods can be. And he then had to, fortunately, later on in the film, lose that weight and be healthier. And all he had to do was go off the red foods and just eat the foods that are indicated as safe, and he did that too. So that's
it is actually hard to gain 30 pounds in 2 weeks, just like I think it's hard to lose 30 pounds in 2 weeks.
It is. But if you just eat your red foods, that'll probably happen.
Yeah, that's pretty crazy. That's an interesting story. I mean, I'm sure you have plenty of stories that people have shared with you about people that have gotten well. I guess what I want to talk about next, and maybe some other story will come to mind, is one of the things we talked about last week was that there's a lot of different testing to identify, I guess, what to eat and what not to eat. Meaning like IG type tests, ELISA tests, gut permeability tests. And this is one of the things that I think is super important of number one, why someone should consider Alcat over the other types of testing and what it's testing differently and how it's testing differently. Can you go into that? I've pulled up my Alcat results on the screen right now. Not that they're important, but people can kind of see what the result looks like, you know, when you're looking at someone's panel.
Yeah, you know, it can be any kind of a food, and sometimes it's surprising as to what it actually is. You had that graphic before of the cells and graphs.
Yeah,
If you,
I'll pull that up.
if you put that one back up there,
Here.
that helps. Okay, so first of all, what is Alcat testing? So with the Alcat, we take a person's blood and get it to the lab overnight. We want to do our analysis maximum within 72 hours of the blood draw. And essentially what we do is we expose the blood to the individual substance, food extract or food chemicals, additives, environmental chemicals, drugs, molds, functional foods and medicinal herbs, and all these different things that people consume or have contact with. And what we're looking for is how the peripheral, meaning the blood, white blood cells, which are the immune cells, respond to that substance, while in the context of the whole blood, meaning there's no separation of the serum from the cells or any of that, so that everything that could be involved in a reaction, meaning what we call cytokines, chemicals that cells release as messengers, serum proteins, antibodies, proteins that are in the blood that are involved in immunological reactions. There are all the other cells that can communicate with one another. They're all present, so we create a situation which is as much like real life as possible. And we put each sample, we incubate it at body temperature for a period of time, and we put each sample that we build that will measure the individual white blood cells. And what happens is we also run a number of samples of the patient's blood without a test substance. And from that, we get a picture or histogram of the size and distribution of the leukocytes. And where you have your pointer right now on lamb, if you were to look at the broken line, if that's visible on the screen, that's what this patient in particular, that's what their blood looks like expressed in a histogram, which again is just a size distribution curve without anything added to it. That's the broken curve. Then when we add lamb in a separate test well and expose the lamb extract to the blood, it creates another size distribution curve or histogram, and then we compare the two. Now in this case, you can see that on the upper part of the larger peak, and that peak are the phagocytes, the granulocytes, there's a big space at the top where there's a number of cells that are missing. So then we took that sample, actually we sent this out to an immunology lab years ago, and looked at the cells when you see that kind of graph. And the picture above that is actually a picture of a cell that has gone through a process which is called degranulation. So basically the cell has reacted to the lamb molecules as though those lamb molecules were a dangerous pathogen. And a number of preformed mediators that are inside the granules, that's why these cells are called granulocytes, have spewed out in an attempt to try and neutralize that. And in the process, there's been the generation of free radicals, which are damaging to other molecules, which again are defense. But in this case, it's defending against something which is ordinarily safe for most people to be exposed to. The next graph, barley, shows another kind of reaction. The cells have swollen up, but they haven't yet gone through that process of degranulation. And that's reflected on that kind of a graph. And then the third one, the negative control, that again is the blood without any substance added to it. You see a nice round neutrophil or leukocyte, which is not reacting. So if we get graphs like that one on the far right, we know that the cells are not reacting to it, and the person can consume that food or that substance. If we get a graph that looks like the barley or the lamb, we know that that's something that's probably going to be problematic for that person. So Alcat is the only test that really does exactly that. And the concept here is that there might be a number of different factors that eventually lead up to the cell's response in this kind of a way. It can be from a chemical that's inside of the food. Let me explain that. A chemical that naturally occurs. All plants contain toxins, natural toxins that they have to protect themselves from pests. So when you eat even a healthy food, broccoli or kale or carrots or whatever, there are natural toxins in there. Like potato has solanine and so forth. And that might be something that you might not detoxify because of your genetics. You don't have the capability to break down those toxins, or you do have the ability to break them down to some extent, but not entirely. And then you get an overload of that. And then the innate immune system, which is what we're looking at here with these peripheral leukocytes, comes into play and neutralizes that toxin. Now, in that case, we call that a pharmacologic reaction. There's no antibody involved. It's directly a pharmacological mechanism, but yet it can have this kind of an effect on the cell. So the difference between what the Alcat measures and what the immunoglobulin tests measure is that we're looking at the cell, and it's the cell that produces the inflammatory chemicals and mediators and free radicals that cause the type of symptoms that we associate with adverse reactions to foods. It can happen with the presence of an antibody. It can happen in the absence of an antibody. We don't really care. And in fact, the antibodies that are usually tested for food sensitivity are there actually to protect us. So they're more of an indication of tolerance than they are of intolerance. Not so in the case of true allergy, where they look specifically at immunoglobulin E, which is very rare in the blood, but that is a good marker of true allergy. And the difference essentially is that a true allergy, the example is if you eat shrimp and your throat closes up, that's an IgE mediated reaction. Hence, looking at the serum for IgE antibodies against shrimp is very useful and informative. But the IgG antibody tests, when you match them up in rigorous experimental settings and feed the person the food in a double-blind fashion in the right way, and see if the test compares with the actual feeding challenge, for the most part, it actually doesn't compare. And because logically, it's just looking in the wrong direction. And there's a long history of that, and I don't want to dwell too much on that point, but just the essence is we're looking at the cells, which are the factors that produce the inflammatory processes.
Do you think that the cell response is more relatable to autoimmune responses, or not really?
Very much so. You had on the cover of that book, the cell graphic.
Yeah, I'm gonna move back up to the top. I have to move this little thingy here. Here.
Okay. So what do you think this is?
I think it's an autoimmune response because you told me earlier.
Okay.
I cheated.
This is a neutrophil. This is the most prevalent white blood cell in the circulation, and it's involved in an Alcat reaction. But this neutrophil has undergone a process which is called netosis, which means neutrophil extension traps. Now the cell is actually undergoing a kind of a reaction that we were showing in the previous slides, but it's going a little further. And what's happened here is that the nuclear material genetic material inside the nucleus, the DNA and the histones that the DNA is wrapped around, the membrane proteins from the outer membrane, the membrane lipids from the outer membrane, and the inner membranes from vesicles that contain toxic chemicals and so forth, have sort of merged together and extended out in these traps that can trap, in this case, a bacteria, a fungi, some other pathogen, a virus. And the DNA, which is now sort of mixed in together with all these other cellular components, is very toxic. And it actually kills these pathogens. And that's a very sophisticated, fairly recently discovered in 2004 at Max Planck Institute in Berlin, defense mechanism. Now, what happens is after this whole mess occurs, there's a need for it to be cleaned up. And there are enzymes called anything that ends in ASA as tryptase, kinase, is an enzyme. So there are enzymes called DNases, which actually break down the DNA after processes like this have occurred. Now, if they're not functioning properly or if they're overwhelmed, you have a lot of what they call cell-free DNA in the circulation. And that cell-free DNA has been positively associated with autoimmune disease, particularly lupus and rheumatoid arthritis, but most probably a number of others. So this is, as I mentioned earlier, I was going to talk a bit about a more sophisticated cellular response, and this is what I have in mind. This process called mitosis, neutrophil extension traps. So we actually, I think you're probably aware of this, we actually, over the years, have been very open about giving access to our technology to independent institutions to study it. So we've had people from the University of London, University of Ottawa, Camerino, Northern Illinois, and Yale School of Medicine, and others look at our science. And then most recently, people from Yale found out that when you have a positive Alcat reaction, you do in fact have a greater amount of cell-free DNA in the fluid left over after the reaction. So there's a very strong likelihood that a food can trigger a cellular response that evolves into this kind of extracellular trap thing, and then the release of cell-free DNA, which through other mechanisms, which are now being clearly understood, promote autoimmunity. The DNA is, cell-free DNA is not always immunogenic. So we actually received, after the Yale study, some research funding from one of the larger biotechnology companies to drill down and characterize if the DNA that the Yale people discovered was in fact pro-inflammatory. And that research is going on right now at Georgetown University and University of Leipzig in Germany. And at this stage, it looks likely that the outcome will show that following an Alcat reaction, there is a release of DNA that is pro-inflammatory and that we now know does drive autoimmunity. So if you ask me, I think there's a very strong link between food sensitivity and autoimmune processes. So it's kind of groundbreaking because it demonstrates in a scientific way that there's more to food sensitivities than just being something of a nuisance. And I think
right, and I think that the amount of autoimmune people, I don't know if it's just my luck that there's so many of my clients dealing with an autoimmune issue. It seems like, I don't know, I mean, maybe I have my own little bubble going on here, but it does seem like that it's super prevalent. And I've heard some people say more recently that, you know, autoimmune disorders, percentage-wise in the population, are higher than ever. I don't know really what to attribute that to. I have some ideas, but I think that, I mean, my experience, I know that, you know, Alcat, as you know, I mean, you know, because you're a CEO, but you know, it sometimes is a thing where people's like, oh, the, you know, 200 foods is, you know, this much money, and it might seem expensive, but I tell my clients that, I mean, I don't know about everybody, but I spend a lot of money on food, good food, organic food, and nutritious food, and grass fed, and all that, but also supplements and all that other stuff. And if you're having a one of these reactions, like a cell explosion to something, and you don't know what it is, I mean, you could be trying everything. You know, there's so many. There's a big thing that I hear from clients is between leptins and blood type and metabolic and paleo and keto and all these different theories, and even theories with anti-inflammatory, like my own story, almonds and lemons and olive oil, is that if you can't stop the inflammatory process, then I think that you can't get better. I mean, it feels like to me that the money, I'm obviously an advocate of Alcat, but it seems like to me the money is necessary to know how to stop the reaction, because I feel like with my clients, if I can get them to do Alcat, and if they'll follow the plan, which is the second part is being compliant, then they have a chance of emerging from whatever they were dealing with. I mean, most people that know my story know I used to be on 9 medications a day. I would not consider myself a healthy person then, and I'm on no medications now, and I think I'm thriving and feel great for my age. So I know it can be done. I mean, I wasn't raised, eating granola bars and stuff. I was like a normal American kid eating Captain Crunch and whole milk. But my point is that I feel like, and maybe you have something to say about this, but I feel like if you can't break that cycle that's causing the inflammation, and now I've learned more from talking to you last week and today, that you can do all these other things and still not get the results that you're seeking because you've never quit the inflammatory response from the food you're putting in your mouth. Does that sound correct?
Yeah, yeah. So yeah, you can even be eating really what generally are considered and are healthful foods, but for you, they're not. And if you don't know that, you're undermining your health entirely. So it makes a world of difference.
Yeah, I pulled up my results again just because, one thing I was doing right before the results I mentioned to you earlier was I was having, well, with the green pea thing, I was having a green pea protein shake with vanilla in the morning. Most days I was having a salad with turkey, boiled egg, some apple cider vinegar. Well, every one of those is on my moderate list. I can't have lettuce, I can't have apples, I can't have turkey. I was having sweet potatoes instead of white potatoes. I can't do that either. So, I mean, just looking at my moderate list and the severe, I was just working against myself. So this brings up my next question that I didn't ask you before, but how often, and when I want to talk about the desensitization and reintroduction, because you told me something about that last week that I wasn't aware of, but I mean, how often do people get Alcat done, like for the foods particularly? Because mine has changed over these 15 years. I've done it 3 times, but the almonds seem to stay there. And I know that we talked about the reintroduction, something may not desensitize. So can you talk about that a little bit?
Yeah. So this whole desensitization means avoiding a food or substance for a period of time and developing the ability to tolerate that again. And what we've seen over the years is that if a person has a test this year and follows the test, 1 year later, on average, they will have about 50% less sensitivity. So that means that you may be able to reintroduce something or you may not. And one of the things I wanted to explain, to give a little context here, and I just was reading the paper, and I know you can't see it because I don't have, oh, there you go. I don't know if you can read that. This is a paper from 1946, written by Theron Randolph and Frank Rolling, allergists, and they're part of the early pioneers in this field of looking at cellular responses to foods. So we didn't invent that idea. That was around back in the 1930s. There were people who found out that if a person had an adverse reaction to a food, after eating that food, their white blood cell count would go down. So that information was already out there. Theo and Randolph particularly is someone who advanced that understanding tremendously, because he, and this all makes sense in a moment in terms of your question, he would sit with patients for about 2 or 3 hours and meticulously listen to the patient's stories about when their symptoms would get better, when their symptoms would get worse, what they eat, what exposures would they have. And he found, after doing this with hundreds or thousands of patients, patterns. And what was really interesting to him was that, I'll give you an example of this. Let's say that a person worked at a printing company and developed a sensitivity or an adverse reaction to the ink. Well, during the work week, they might have their symptoms, but they wouldn't be flaring. Then they go on a vacation for a few days, or even just a long weekend, and their symptoms would temporarily get worse. So he figured that he saw that pattern enough to realize that there was a process going on called masking. So that if a person, masking, M-A-S-K-I-N-G.
Okay, masking, okay.
Yeah, so that if a person oftentimes will crave those things that they're sensitive to, because it will, for some reasons that we're not completely clear right now, it will actually somewhat suppress the symptoms. And then when the person goes completely away from that offending substance, a food or a chemical, they may actually feel worse for 4 days. And that seems to be about the right time, 4, maybe 7 days. And then they feel better than ever. So they go through kind of like, you can call it a detoxification. I would prefer to call it an adaptation.
It's like a withdrawal symptom.
It feels like withdrawal symptoms, and it kind of is withdrawal symptoms. Some people say that, well, when you're exposed to food all the time, you make a lot of antibodies, and when they're complexed together with the antigen, they're removed by special cells called macrophages and monocytes, and it's silently done. There's no symptoms and so forth. And then you stop exposing yourself to that food, then you have an imbalance of antigen and antibody. You have still the antibodies that have a longer half-life and less antigen as it's cleared. And that produces symptoms like serum sickness. And that could be one of the mechanisms that are associated with it. But it means that it's probably, again, something, the immune system, just like any other part of the body, but I would say, all systems are very complicated, but the brain and the immune system together are probably the most complex. And the immune system has such a challenging task. It has to protect us, but at the same time, not react against things that are safe. Now, we used to say, self and non-self, but that's sort of been overridden now. I'd say there was an NIH researcher, I forgot her name right at the moment, it'll come to me, who, a brilliant lady, she studied antigen recognition of lymphocytes at NIH. She used to be a Playboy bunny, and she used to, during the day, work as a waitress at a university town, and hear the scientists come and talk and ask these really deep questions. They have such a deep curiosity about life science. And they said, you really ought to go and get your PhD, and she did. And then she finally rose up to the point where she now has her own laboratory. And she came out with the concept of what we call DAMPs, or danger-associated molecular patterns, which are things that cells have on their surface or inside the cell that recognize factors that are dangerous. And she said, look, this self-non-self model is too simplistic, because we're exposed to non-self stuff all the time. The immune system tolerates it. It's a question of danger or not danger. So the immune system has to discriminate what is dangerous and what's not dangerous. And it could be something external, like an environmental factor like dog dander or some pollen or food, or can even be something endogenously. So, and the example of that would be, for example, DNA that leaks out of the cell for reasons that we were talking about, a cellular reaction leading to this very advanced kind of defense pattern. So it's a question now that the immune system has to recognize what's dangerous and what's not, but yet all the time be vigilant and react quickly to something that in fact truly is dangerous. So that's walking the line, if I ever heard of walking the line. So when you start throwing so many different things at the immune system, which I think we're doing much more now than we have in history because of pollution, you're making it more challenging for the immune system, number one. Number two, the immune system, as I mentioned, comes into action when there's toxicity. Now, we have detoxification pathways, but those detoxification pathways rely upon certain cofactors. Now, those cofactors are what we call specific micronutrients. And if we're eating food that doesn't contain as much nutrition as it used to, and that's a fact, when we have 99% of our agriculture today is industrialized. And so we're kind of keeping the soil alive with artificial fertilizers as opposed to growing food in a way where we rotate crops and we let the compost there rot, and there's a lot of earthworms doing their thing and recycling matter into forming a lot of essential minerals and vitamins that the body needs. And if the soil doesn't contain the nutrients, then the food that's grown in that soil likewise does not. And therefore we're imposing on the one hand more challenges to the body, and we're giving less support to the body's detoxification pathways. So it's no wonder that there is, and that's just a theoretical model. I haven't proven that myself. And I guess you can, if someone's smarter than me would look deeply into all the literature and pull it all together, there'd be a lot of tremendous support for that model. But it's no wonder that we see, as you're seeing in your practice, more and more challenge with things like autoimmunity and sensitivities or vague symptoms that really defy any specific diagnosis because they're not really a disease, but they're a disorder. So back to your question, the desensitization. Well, if you, the other factor is intestinal permeability, proper digestion. So if your gut microbes are well balanced and you have the right microbes there, not the wrong microbes there, they help. They carry out a lot of functions. Our gut microbiome is like its own organ. It's about 2 kilos and has more genes as an aggregate as we ourselves have. It's very important to us. It helps in digestion. It helps train immune system cells to function properly. And that's why we have now what we call the hygiene hypothesis, where we live in such a sterile, some people live in a sterile environment and develop allergies because their immune system hasn't been trained to tolerate environmental things. Kids grow up on farms with animals, they eat whole foods and their immune system is better, they have fewer allergies and so forth. So all these things play together overall to create how we survive and thrive in our environment or don't. So over time, if you live healthy, exercise, manage stress, curb inflammation, get adequate nutrition, exercise, which is good for absolutely everything, you're going to recover and you're going to be able to thrive in the environment much more so than if you don't engage in that kind of healthy living. And then, of course, back to the gut is very important. Some foods, this is where gluten comes in, because gluten is certainly one food. It's not the only one that can induce a sort of compromise in the function of the gut by inducing what's called in the colloquialism leaky gut, or medically speaking, intestinal permeability, which not all scientists believe in right now, but more and more of them do. And that means that the gut actually doesn't perform the same barrier functions that it's supposed to, keeping the junctures in between the enterocytes, the gut cells, tight. So if you have factors, and gluten seems to be one of those, that causes a breakdown in intestinal permeability, the tight junctures loosen up, you get what they call the perfusion of not fully digested food particles going into the blood circulation, which if they had been fully digested and broken down would not be immunogenic. But when they are in a macromolecular state where they're bigger, they might look more like a pathogen and activate the immune system. So we see that sometimes, and I think that might been an issue with yourself, that with your lemon and your olive oil, there's some gut permeability issues, and that was leaking into your system, and it was just too much. And some of the natural components of the olive oil and the lemon, which normally is quite healthy, might have overwhelmed your detoxification pathways. And then your innate immune system, that's the first line of defense, which the Alcat measures, is called into action. And then that's good, except that it also produces these bystander damage through the breakdown of the cells and release of toxic mediators that cause inflammation and all manner of possible imbalances, even neuroendocrine imbalances, and can affect our mood and certainly create what we call type 3 immune mediated problems like arthritis and so on and so forth. It's very, very, very complex, but the thing to focus on is avoid the triggers, live a healthy life, heal the gut, eat nutritious food, and then everything else that contributes to a healthy lifestyle. Alcat's foundational for helping to find out which are the food triggers. And, as you said before, if you don't do that, then you're undermining whatever else you're doing. So that's important. All the other factors are also very important. So they have to be addressed. When they are, sensitivities decrease. Now, we don't know because food sensitivities are multifactorial, and that relates back to your first question. That's why we test a cell. At the end of the day, excuse me, it's a cell that produces the symptoms. If the cell reacts, probably you're going to react if it's a strong reaction. If the cell doesn't react, I don't care if there's antibodies there or not, you're not going to have a reaction because the factors that drive their reactions are just not coming into play. So if you look at this, some of the things are related to gut integrity, some to detoxification pathways. Some of these things are related to our genetic makeup, and therefore we may always have our whole lifetime a sensitivity to almonds, as in your case. We call that a fixed sensitivity. Some of the other things are cyclical. In my case, if I eat too much lettuce and soy and wheat, I'll have a reaction. If I eat it moderately, I can tolerate it. So you don't know where on that spectrum you're going to be. So the only thing to do is to retest or just experiment. But the experiment, one must keep in mind that the reactions could be masked for a period of 3, 4, 5 days. So if you're going to reintroduce, you introduce one food one at a time, and you only do that for a week and see how you feel. And then the next food, because if you every day reintroduce something new, and on day 3 or 4, you feel your headaches coming back, you don't know whether it was food challenge number 1, 2, or 3. So.
Right, I'm glad you said.
So I think the program might have frozen up a little bit. Can you hear me? Hmm. Huh. Huh. Oh, okay. Oh, okay. Uh, okay. Sure. Thanks. Huh.
There we are.
How you doing?
Yeah, okay, so it says being recorded, so this might be a part one, part two, but it just went right back where we left off, I think.
Okay, great.
Yeah, so thank goodness. Okay, so let's see, the last thing I was gonna say back to desensitization was just what you mentioned. I think the last thing I want to ask you on that topic, just to be clear, and this kind of came because a good friend of mine knew I was going to talk to you today on this interview. And so she re-asked the question one more time. So I want to ask it for anyone that still has a question about this. If someone has a known reaction, like to black beans, and they show up on the Alcat results in the green, like neutral, but they know they have a reaction to it. You and I talked about this last week, and they're having like an IgE response. So I told her kind of what you said, because a few people have mentioned this to me, and then she said, well, wouldn't that known like very strong reaction to black beans still cause an inflammatory response that would be picked up? And I know that you said if there was a response, then it would lower the white blood cells, which I think is affecting the immune system and all these other things. But is there any time, I guess my question is, why would that happen? Or can you recommend like a certain specific situation where you might say, okay, someone should do Alcat testing, but they should still get an IgE or ELISA type testing? So can you answer that?
I think it's a really good question. So yeah, I wouldn't go for an IgG. I don't think IgG is really valuable. In fact, as I mentioned, I don't know if I did mention this, but years ago, 3M made a huge investment in buying an IgG test, and then they played with it for a while, and then they gave it to some university to study, and they found out when they really drilled down and look at food challenges that it didn't really correlate, and they abandoned it. But IgE, yes. So IgE is a really interesting thing. It's a great story. Um, it was discovered in the mid to late 60s that the factor that causes allergy is IgE. So going back into the 30s, there was two allergists named Prausnitz and Kutzner. One of them had an allergic reaction to fish. And if you did a scratch test, he would show up positive. He would have a wheel and flare reaction. The other partner did not have that reaction, but they took the serum from the allergic patient and they injected it into the skin of the non-allergic patient and then did a skin test and scratched the skin with the fish allergen, and sure enough, it produced that. So there was a passive transfer of an allergic reaction. They didn't know what it was that was in the serum. They knew it was a serum factor. They called it reagent. And then, cut the story short, in the 1960s, they discovered a molecule in the blood of allergic patients that they didn't know what it was. They found out it was IgE, and then they correlated it with asthmatic blood. And there was cooperation between people at Karolinska Institute and University of Colorado, the Ishizakas, and a team in Sweden. And they found out that IgE was the reagenic antibody that would cause what we call a type 1 allergy, or now an IgE-mediated IgE-mediated reaction. And they created a test called radioallergosorbent test to quantitate allergen-specific IgE. So the thing about IgE is the reason it took so long to discover is because there's not much of it. And when it does occur, it can cause that immediate type reaction. It's the same kind of reaction that is associated with our immunological defense against worms, helminths. Now, if you can imagine, you have a worm infection and you have a white blood cell. The worm is so much bigger, it's thousands of times bigger than the cell. When that reaction occurs, those small cells, which in this case are other kind of white cells called basophils and mast cells, they spew out histamine very strongly to attack, and eosinophils also to attack these big dangers. That creates a dramatic effect. Now, when a food triggers that same kind of reaction, it's very dramatic and it's very pronounced, and it's quite rapid. It's quite immediate. So you don't usually need a test to identify that, but having a test is not a bad thing. Now, the thing is, IgE is not very prevalent, and basophils in the blood, that are the cells that are responsive to the IgE, are very small in number, maybe 1 or 2%. And so the Alcat doesn't pick those up because it's too small of a signal for what we're doing. So yes, one could do an IgE test alongside of an Alcat to cover for those things. And that's the reason why people sometimes say, but wait a minute, I eat crab and I get my throat closing up, but the Alcat may or may not, most likely will not show that. Well, the Alcat isn't intended to show that, nor claimed to show that. So yes, of course, if you know that, then you really don't need a test. But if you want to be on the safe side and confirm it, doing an IgE test makes good sense. I wonder if we can continue this later on, because I ran up against the time factor right now.
Yeah, I think we're good. I think we covered most of what I wanted to cover today. I guess maybe we can just close with well, I think we covered most of what I wanted to get across. So maybe if there's something that we didn't, I can just type the information under the video, if there's something that we didn't. One thing that we didn't talk about, but I can just attach a link, is to the other things that you guys do besides Alcat. And I guess, do you have like 1 more minute, or do you have to go like right this second? So just one last question, and this is more an observation. You guys have been doing the cellular nutrition assay. Have you noticed that people that have more severes and moderates have more food deficiencies, or have you not seen a relationship? I'm just wondering if you've seen a correlation there.
We just submitted a paper on that, and the answer is yes. And hopefully that paper will be in publication fairly soon, and go on to a relationship between food sensitivities and nutritional deficiencies and antioxidant status and other lifestyle factors like smoking and how that impacts on food sensitivity and nutritional deficiencies. Pretty interesting stuff.
Well, yeah, it makes sense if the inflammation's there, that the digestion, the absorption, the uptake would be affected. That's just what I was thinking without knowing what the research would say. That's kind of the thought process with the healthy gut in the first place.
Mm-hmm. Yeah, and the healthy liver as well.
Okay, well, Roger, I really appreciate your time, and we've been on for a while, so I think the information is going to be great. I think people are going to love to get to know you and meet you and know more about Alcat from a more scientific perspective than what I can probably offer. So really appreciate your time.
Thanks so much for having me. It's been a pleasure.
All right, thank you.
Yeah, take care. Have a good evening.
All right, if we missed any questions I need to know, I will send you an email. Thanks.
I'm here. Take
All right,
care.
bye-bye. All right, bye-bye.